Yes. Yes.
Great. Thank you. So I have the honor of introducing you and kicking off our monthly computer graphics invited lab talk series. And I have, as I said, the pleasure of introducing Jan Aerts. Jan is a professor at KU Leuven. I know him for quite a few years now because we're working in the same field. And it's always fun to meet him and spend time at conferences. Jan has an interesting vita because he switched fields in between. So he has a background in omics and bioinformatics originally, and he actually worked on large sequencing projects. And during his PhD, which he did at University of Wageningen in the Netherlands, he worked on the chicken genome sequencing project. So if you remember, the large genomes have been sequenced like one by one, and the human genome was like one of the most complex ones, of course, but Jan worked on the chicken genome sequencing project. And then he switched fields, and his focus now and in recent years is on data visualization, visual analytics, and also data analysis. Before he became a professor, he was also a researcher at the Wellcome Trust Sanger Institute near Cambridge, UK. In previous years, he was a professor at the University of Hasselt, and now he moved actually back to KU Leuven, where he has been previously as well. And Jan is going to talk about biological data visualization, and the title of his talk is From Complexity to Comprehensibility, an integrative view on biological and agricultural systems. And before I give the word to Jan, so welcome everyone who is here on the Zoom call. We have a hybrid setup, so some people are here in the room with us, but most of the people are here online. And for all the seminar students, just as a reminder, please turn on your cameras and use your real name so that we can count your participation. And with that, I'm looking forward to your talk, Jan.
Yeah, thank you. Just checking you can hear me?
Yeah, perfect.
Yes, okay. So thanks for the introduction, Mark. Yes, so indeed, so my background is in bioinformatics and genomics. As you mentioned that, on the first slide, you see that image, that's a picture photograph we took from a 3D object that we created, that's actually based on the chicken genome. That's actually an art piece, data visualization art piece based on a chicken genome. So yeah, thank you for the privilege to talk to you. It's really interesting to talk to this to this type of people as well. It's really good initiative to have these types of talks. I think we should introduce that in our department. Yeah, so in this talk, basically what I want to do is basically explain why I do what I do, and go a little bit into the types of analysis that we try to do as well, that we develop. Okay. So going from complexity to comprehensibility. And basically what I want to start with is, as some of you know, there's quite a few data visualization challenges, visual analytics challenges each year. And one of them is the VAST challenge, so the Visual Analytics in Science and Technology challenge. And I want to show this as as an example of why we need this, right? Why do we need data visualization? This is a challenge from 2021. So a competition, and the story is, it's a fictive story. There's a fictive island south of Greece, and on that island there is a company. Let me check. Can you see the mouse?
Yes.
Yeah, okay, yeah. There's a company on that island, and that company is really clashes with environmental groups. Well, the other way around, the environmental groups clash with the company. And at a certain point in time, basically the C-level employees of that company go missing. So the CEO is gone, the CFO, the CSO, they're all gone. And the question is, okay, what happened, right? So were they abducted? Did they flee with money? Who knows, right? And that's basically the question, what happened? The data that we had for this, like, again, this is completely fictional, is a tourist map of the islands, where there's a company and then some roads and cafes and restaurants. Then there's a shapefile actually with streets on that island. Car assignments, so company cars, I think there were 30 of those. Okay, each car, who is registered to that car. For each car, we had 14 days of GPS tracking data. We had credit card transactions for each of the, well, for many of the different restaurants and cafes and pubs and so on. So what amount was charged by whom on which credit cards, and we also had loyalty card data. And that's the information that we had. The trouble is that there's no way of integrating this information algorithmically, right? You could use statistics and machine learning or just regular data transformations to map things together, right? So you could say, okay, there's a certain transaction at a certain point in time, at, let's say here, Bruce being served. At that point in time, there's a transaction. Who we see, there's a car that is standing still, really close by, and it's this car, so that person will be the one paying. But there's several issues. First of all, the GPS tracking data were really, really bad. So cars would be driving in the Mediterranean. That does normally not happen. Also, if a car is in a certain location, doesn't mean that that person is driving the car. If you pay something, it might be that you pay for multiple people and so on. And we really needed to use data visualization and visual analytics to actually come and solve this big problem. The second thing I want to make clear here is that the question was an open question. So what's going on? It's not that we have very specific hypotheses to test. And that'll come back later. Well, if you look at how research has been done in the last centuries, right? So the reductionist approach in scientific in the scientific method has been very, very successful, right? So breaking things up into smaller pieces and then trying to understand the smaller pieces, and basically the system itself is a sum of those pieces. But at some point, you run into a wall. Reductionism doesn't work anymore. And that's why we have to look more like at a holistic approach for analyzing data, because you have these emergent properties, right? So, like a single neuron in a brain will not think about that person's grandmother, right? Or a single H2O molecule is not wet. It's a collection of H2O molecules. So you really need to look at many things at the same time. And that is especially true in biology or any biological systems, biosystems, where everything is connected to everything else. So this is a graph from a paper about sustainability of water, land, food, and food resources in Africa. And as you see, everything is connected to everything else. So the crop yield has an effect in food availability, which has an effect on population growth, which has a feedback loop to food availability, and so on. So these systems, there's many feed-forward and feedback loops, and those feedback loops may be reinforcing. So basically, they reinforce each other, or they might be balancing. So they keep each other in balance. Right? So completely opposite effects. So systems are very complex, and that's kind of related to what we see in biology as well, in omics. So if we have omics data to analyze, there's genomic data, transcriptomic data, proteomic data, metabolomic data, and so on. And we want to integrate all those things. Now, how does this all fit into that holistic approach? Basically, what we try to solve is the wicked problems. The wicked problem, that's a term that's been used, I think, first in 1973. And that those are really very complex problems. What is a wicked problem? It basically has three properties. One is finitude, which means you will not have enough resources to test, to solve the problem. For example, you can only solve a problem, or find a solution if you have the DNA sequence of every individual who ever lived. That's not happening, right? A second characteristic of a wicked problem is that it's complex. So you have several feed-forward loops, feedback loops, partially nested hierarchies, and so on. And the third one, which is very often overlooked in data analysis, is there's a very important value for, input from the user needed. So any solution depends on the values and the norms of the individual who looks at a problem. For example, let's say you look at trying to solve crime in the street, right? So you try to solve crime in the street. What you can do is you could put more policemen on the street. That's something you could do. You could arm the policemen who are there, or you could actually take away the arms from the policemen, because a criminal will be less likely to attack someone who is not armed, right? Or an extremist, you could just give free televisions to any potential thief, then they will not steal televisions anymore, right? So it really depends on who's looking at a problem, what the solution could be. So, but these things we cannot really solve because of those reasons. So we have to make them simpler towards what we call complex problems. So, for example, how do we prevent climate change becomes how do we reduce CO2 in the air? But actually research ends up being something that you can test. So what we call data engineering problems. So how do we improve the efficiency of a certain engine with 25%? And the big difference here is that we go from open questions to closed hypotheses, right? And statistics really lives here, whereas what we try to do, we try to live more in this realm, right? Asking the questions. Well, we start with the question, we help people develop hypotheses. So, and that's definitely necessary in agriculture and in biology, because, for example, looking at data in agriculture is very difficult to integrate those data. So you might, for example, have a selection of fruit cultivars, have different levels of omics data. You look at the color of the fruit, the yield of the fruit, the taste of the fruit, chemical analysis, interviews with users, economic data, and so on. That's all very difficult to integrate. So in the traditional approach, so the statistical approach, basically what you do, let's go back here, in statistics, having a system like this, you could check like what the effect is of, let's say, labor force availability on crop yield, the crop yield on the net farm income. But you always have to just look at one slice of that very complex system in statistics. We try to look at everything at the same time. Because if you hide complexity, you end up having issues. I'll show the issues in a second as well. But what complexity exists there? Well, where do we get complexity? One of them is, for example, in the parameter space. We're now currently doing a analysis where we look at microbiome research. So the microbiomes are everyone, so that there's microbes everywhere, on our skin, on every single surface that you have, in your gut as well. And actually in the bioinformatics pipelines to analyze those microbiomes, just changing parameters has a huge effect on what results you have. And the problem is that many researchers don't know what parameters to play with. And the second one is a source of complexity is the sampling itself. If you want to take a microbiome of the soil, for example, say you have a field, and take a soil sample and want to measure what microbes are in there. Well, what if you had taken that soil sample 5 centimeters away from the original location, or a little bit deeper? What if it had rained the day before? What if there's the root of a tree that is in the neighborhood? They all have an immense effect on the microbiome in that sample. So it's very difficult to actually get knowledge out of this. And we do have to embrace that complexity, because if we don't embrace that complexity, what do we get? We get misrepresentation of the system. And a very important thing is limited insight, right? So we can make decisions, but those decisions is on incomplete data. They are biased and so on. And a fourth one that I mentioned here is reduced resilience to disturbances. So if one parameter, so to speak, changes, then you might get a different result and you don't expect that. You don't know what's going on. So how do you tackle this? Basically by involving multiple people, looking at something as a continuous process in the analysis instead of collecting your data, sitting behind your computer for a while and then coming with a quote-unquote answer, but more continuous improvement. And putting things into context. That's one of the keywords that I also use as well, putting things into context. And for that, we need to combine computational strength with automated analysis, which of course is visual analytics. Many of you will know what visual analytics is, but for those who don't, so basically in visual analytics, you try to combine data visualization, interactive data visualization with machine learning or AI, so that you basically get to insights in the end. I mentioned AI, artificial intelligence. What I do, I also call it AI with augmented intelligence, which is basically combining these two as well. So augmenting the capabilities of the user. Now, if we compare visual analytics with statistics, I hope none of you are hardcore statisticians, because I like to take some shortcuts at some places, but still, for those people, I'm sorry. But let's say you have as a researcher, you have a certain question, you try to get in some direction, right? And statistician comes in, there's a hypothesis, and I sit behind their desk for a while doing the statistical analysis. They may do some p-hacking or not. They get a p-value, and that p-value may lead to insight, even though a p-value is not the same as insight. What we do is, with visual analytics, you start earlier when you still have an open question instead of a closed hypothesis, right? Not something that hypothesis you can check. A question you cannot check, you try to understand it. So we start earlier, and then it's a very iterative process. So always together with the experts, and you get small insights along the way, but also because of those small insights, basically what that expert wants to look at, that changes over time, because they have this renewed view on their problem constantly. So what I want to do in the remainder is look at both of, well, the visualization aspect and the automated data analysis aspect, right? So basically what we try to do there in each of those. So the visualization aspects, there's different things. If you compare data visualization with data analysis, let's just look at data analysis. A tool like Excel is really, really useful. Excel has a huge number of users, but the types of questions you can answer with Excel are quite generic, right? Quite general. The moment that you start looking at biological data and microbiome data or other very complex data, then Excel will not cut it, right? Then you have to use R or Python to write a custom script. So then compare that custom script, that one script versus Excel. So you're in this realm here for very specific questions. But of course, the number of users is very low then. It can be just 2 or 3. So just like that's the case for data analysis, that's also the case for visualization, or what we call visual encoding. So the very generic plots like a bar chart, scatter plot, and so on, they're really, really useful, but you can only go this far with those. So the moment that you go into very specific types of data, then you have to develop new visuals. So this one already existed. That's the Sequence logo from 1986. But these are two that we developed ourselves for looking at microbiome data, this one. And the bottom one is a redesign of the Sequence logo at the top, looking at multiple sequence alignments of DNA or protein sequences. And here we have some other examples of custom designs. All of these are ours, except these two at the bottom here. circles, and the hive plot are from Martin Krzewinski from Canada. Coming from omics, most of the examples come from omics as well. So looking at genomic structural variation, for example, bits of a chromosome that are cut out and then moved to some other location, for example, stuff like that. And what is very important in that area is actually design. So there's different things we do on the visualization specific side. One is the visual design. So actually developing the new visual, what should it look like, together with the experts. So basically, it's a very iterative process again, trying to develop new ideas for visualization. And this is a picture in one of the sessions that I made. I gave a workshop once for the UN, and this was in Italy, and this was a picture during one of those sessions, really discussing a lot of things and pen and paper. So the visual design is an important part. Then interaction design is an important part, but less so, but still. And then scalability is another part that we look at. Scalability, more scalability of perception than scalability of data processing, even though that's also something that we sometimes need to look into. As an example of that iterative process, so like I mentioned, so the Sequence logo from 1986. This is another example of a competition that we participated in. So basically, this was the starting points, and they wanted to have a redesign where you could actually compare. Let me have a quick look. Yeah, compare what the difference is between the two bottom ones, gram-positive or gram-negative bacteria. This is one protein, but then in multiple bacteria. This is in all the samples, all the bacteria. This is gram-positive and gram-negative bacteria. And the brief of the competition was, okay, develop something where it makes it easier to actually compare the two bottom ones. And this shows you how basically you start from something and you redesign. It's not that you start with something and then all of a sudden you come up with the final design. It does not work like that. It's really iterative, which is completely different from many machine learning of statistics approaches where you have to define beforehand, okay, these are the things I'm going to do. And this, especially in statistics, this is the test I'm going to do. And of it needs to be written down. It comes from a protocol even. And you just perform what is in there. Here you really see what goes on, and then you try to improve as you go on. But in the end, having these things, you end up creating like an interactive user interface to explore that data and to get insights from that, right? So that's basically, to wrap up the visualization part of what we try to do, which is we have complex data, and we want to combine multiple things together into contextualize the data and look at multiple modalities at the same time, which means that a scatter plot doesn't work anymore. So we need to develop new visuals in the specific fields that we work in. Now, for the automated data analysis part, we do regular machine learning, mostly unsupervised, clustering and dimensionality reduction. But the core of what we try to do is based in some way on topological data analysis. So that may not be familiar to everyone, but the last couple of years, actually, I see it's getting more and more traction. And the premise of topological data analysis is that data has shape and shape has meaning. And then as the company, ISD also says, well, data has shape, shape has meaning, and meaning has value, right? So what is topological data analysis? Consider this point cloud. If you would run a clustering program on this data, it would give you 3 clusters, right? That's 3 clusters, but I would argue there's more going on than just 3 clusters, right? So here we have something with a bifurcation. We have a cycle here, and here we have 4 different what we call flares sticking out. For example, this could, for example, be let's say a a flight from an airplane, if you take that flight and you chop it up in 1 minute segments, for example, if two of those segments are similar, then they're close together. So you can see that, for example, if an airplane flight, if it's cruising, that's this area. If it's taking off, that'd be this, and if it's landing, it could be these points. Right? This is something that you very typically see in biological systems, circles. I don't know if I have that slide. No, I don't have that one. I should have included it. But for example, you can see that, let's say you have blood values. So you take the bloods and then do analysis, and you see how many red blood cells, white blood cells of different types, and so on. You can measure those. And there's a really nice example where we see that in mice, but also in humans, if one point here, right, if that's a mouse, right, and they measure different things in the blood, like I said, the platelets, red blood cells, white blood cells, and so on. So healthy mice are very similar, and if a mouse gets sick, actually they start moving in this direction, and then they're ill. Right. So basically, you see that certain values start changing, and then they have changed. But when the mouse actually gets oh, right. Apparently, if I do a thumbs up comes on my screen. But if a mouse gets better, then it's not the inverse of getting ill. Basically, it follows a different path. There's different ways to actually get to that underlying shape of data. The most well-known one is called mapper. So what happens in the mapper algorithm, consider like a three-dimensional point cloud of a hand. This could be like millions of data points. Right? What that algorithm does is it looks at some, what I call a lens. In this case, consider the horizontal position. So we color according to the horizontal position, and then we basically make different bins. So these are the data points split into different bins. Within each bin, you do a clustering. So the first bin, we have 3 clusters. So we have 3 clusters. Here we have 4 clusters, and these are all connected. So this is one big cluster, right? One big cluster. But when we take the bins, we actually make sure that the bins overlap a little bit. So some of the data points here are the same as the data points over here. And having our clusters, if we see that some of the data points are in common between 2 clusters, then we draw an edge between them. So based on this point cloud, it could be millions of data points, we end up with a network of how many nodes? 11-ish, something like this. And this is basically the shape of that hand, right? So we also developed our own method called STAT, Simplified Topological Approximation of Data. I'll come back to that later as well. Actually, not later, now. Basically, our own methods works a little bit differently. In this method, in Mapper, each node in the network is a cluster of data points. And there's a lot of different parameters you have to choose, like what is your lens? What direction do you look at? What's the bin size? What is the size of the overlap? What is your clustering algorithm? And so on and so on. And we want to actually get rid of as many of those parameters as possible. So the method that we developed some years ago, well, Daniel Arcadi in my group developed, is you take a high-dimensional data space. In this case, high is 2, so 2-dimensional data space with points organized like this. You create a distance matrix in this space, right? That could be like cosine distance, for example. Based on that distance matrix, you create a fully connected graph and then the minimal spanning tree based on that weighted graph. And then what you do is actually we have the distance matrix of these points, but here we can also calculate the distance in the number of steps, the length of the path between 2 steps, right? So for example, between this point here, well, let's say between this point here and this point here, it's not that distance is not that far, right? But here, actually, you have to follow all this way down and then all the way back up here. right? Because this connection is not there. So what we then do, we have this minimal spanning tree, and we start adding one by one, well, not one, couple by a couple edges. And very quickly, actually, a link will be made between this end and this end. So we get this shape back. And we keep on doing that. And for each of these, we create a distance matrix, again, based on this length. So we get distance matrix based on this length, and we calculate the correlation with the original distance matrix in high dimensional space. And basically, that's what this plot actually shows. You take basically you have the highest correlation between the two. This is your final network. As an example, Daniel was from Barcelona, and he ran this data on a traffic data set from Barcelona. So where each node is a day, it's for a bit more than 1 year. Each node is a day, and the bigger the node, the higher the traffic density. The color of the node has to do with the day of the week. And the connectivity, well, it's if they are similar to each other. And we see the weekdays here. The Fridays are a bit different than all the other days. I'll get back to that. Then we have the weekends, so the Saturdays and then the Sundays. And then we see, for example, the 25th of December is not, well, it's a weekday, but it's very, very different from a regular weekday. We also have these days, which actually Dino could actually explain why they are in between. But I'll get back to this later as well. But we see that it clusters quite nicely the different types of data. We also looked, of course, at how accurate is this method by creating shepherd diagrams. So looking at NMDS versus the original data, our method versus the original data. So basically, the distances were quite well maintained, especially if you combine that. Well, if you look at the TSNE or UMAP, that's well, that they perform much worse. Now, this is the distances in the graph, underlying graph. These are the distances based on what type of layout algorithm we use. So this is after it's put into 2D space on the screen. This is the original graph. Well, the topological data analysis is really strong. And as an example, I'll show you this. I don't know if you know this data set or this analysis, but the NKI data sets, that's the Dutch Cancer Institute data set, is patients with breast cancer. And so this is mapper. So each node is a collection of patients. And it was, quote unquote, known that patients who have a low ESR1, estrogen receptor 1 expression of that gene, that they would not survive. That I think for about a decade, that was like what was known in from literature. But using topological data analysis, I was hiding something. Actually, they found this is a paper from 2013, about 10 years after the dataset became available. There's a subgroup with low ESR1 expression in the people who survived. That was completely unexpected. Even more so, if you take all these patients and you do a clustering, hierarchical clustering or PCA, and you select these patients, these ones, you cannot actually find those back. So they're like spread out. If you look at clustering or PCA, you cannot identify that subgroup. So topological data analysis is really strong there. Somewhere else where we have actually applied this is in sleep apnea detection. So sleep apnea, where someone stops breathing during sleep. It's quite high dimensional data as well, multimodal data. But this is just looking at a signal of chest volume, so basically breathing out and breathing back in. And in periods where someone is breathing naturally, we really see this is the raw signal. After topological analysis, you see breathing in, breathing out, breathing in. Again, that circle, right? The moment that you have a bout of sleep apnea, when you stop breathing, then that circle disappears. And we can actually do analysis with that. Another example, this is not by us, but something that we will be doing in the future probably as well, is a project AI for AI, so artificial intelligence for artificial insemination. where they basically put cows full of Fitbits, measuring movement of the different legs of the neck and so on, location sensors, also measuring when they would go eat, things like that. And based on that information, actually they could build a network like this, seeing what the cow basically was doing, and then if they're jumping, grazing, walking slowly, but especially when they're cow mounting, that's a signal that they're ready for artificial insemination. So I mentioned that here this signal that appears that disappears. That is something that actually you can use in automated analysis as well. For example, looking at grape clusters, that lens that I mentioned with the hands going horizontally. Here, if your lens is the like the geodesic distance to the base, then a certain analysis of the grape cluster shows you this plot, which basically indicates that all the little branches are about the same length. That's what this tries to show. Another something else here is on the right, looking at an orange, at the size of orange, at different intensities of X-rays. And in that sense, you can actually, at different intensities, you can identify different parts of the orange, like exocarp, where the pith is, the rind, and so on. And you can get a lot of different information based on the persistence diagram. But we mainly use it for the visualization. Now, one of the strong things about topological data analysis is that you can put domain knowledge into it. If you think about the PCA, right? If I run a PCA or Mark Streit runs a PCA, we'll get the same thing. Well, normally. These need the same thing, right? But here you can really put domain knowledge in. So for example, let's say you have two patients, and we have the diagnosis for each patient, so a list of diagnoses for each patient. And this is actually from a data set from an ICU, from intensive care units. And different patients have different lengths of diagnosis lists. And the first one is the most important, and it gets less and less important. But then, okay, how do you assess how similar these two patients are? Then we can develop our own distance metric to actually say, okay, this is what I consider two patients to be similar, right? And based on that data, and trying this out, we actually did this little experiment just looking at patients that had somewhere in their data alcohol withdrawal delirium. You have to pick something, right? So you pick alcohol withdrawal delirium for the ICU. And we get this network where community 1 are patients that have alcohol withdrawal delirium as their main diagnosis. This community here, the orange one, is people who came in also with alcohol withdrawal delirium, but the main diagnosis was a skull fracture. So basically, they were so drunk that they fell on their head and they broke their skull. And the purple community are patients who also fell down but didn't break their skull, they just had a concussion. So that's one way to actually put domain knowledge in by changing the distance metric. Something else you can do is actually use a lens. So consider this data set where we have 3 dimensions. We have X, Y, and color. And well, no, let's look at this first, the table first. So let's say we have samples and we have 10,000 parameters, and 2 of those samples have exactly the same value for all those 10,000 parameters except for 1. Well, of course, PCA, TSNE, whichever dimensionality reduction method would say, okay, those are basically the same, right? There's only 1 parameter that's different. But if I say, well, actually, that's an important one, how do you get those? Well, how do you pull those apart? So here we have X, Y, and color. So normally, if you would run a TDA, you get a network like this. But here you go from yellow, there's blue and red here, and there's yellow again. But if I say, actually, I want to really get the red and the blue not together, what you can do is you get something like this. You can say, actually, this is one that we want to pull apart. And that opens a lot of different possibilities, okay, because you can look at the same data set from different views, right? Well, coming back to the data in that traffic data in Barcelona, this was a plot that I showed before. But of course, I also already mentioned that something here is going on with the Friday that's different than the other days. And if you look at the traffic over the day on for all the days, then on Friday about 4 o'clock, there's a traffic congestion peak. So we use that as a lens. And instead of this plot, we get this plot. What happens is basically that this is pulled apart and this is pulled apart. And then you start looking, okay, why are these two pulled apart? Why are these now not similar anymore to these ones? Because they were before. And looking at the data, apparently these days, the ones with the yellow numbers, there was a renovation going on one of the main roads in Barcelona. That's information that actually you can find based on this. Then briefly, some things that we are working on at the moment. So topological data analysis is like what we try to do, and you do that in many different ways. For example, if you look at HDB scan, a clustering algorithm, so density-based clustering. If you look at the HDB scan, it would basically identify, in this case, 4 clusters and a lot of noise. But coming back to the picture that I showed in the beginning, well, there's more going on in the orange cluster than just being one cluster. So Jelmer Bot in my group, he's developed Flask, so flare-sensitive clustering, which actually is able to identify the different clusters themselves. And we just submitted a paper on that, and this is an example of some test data as well. So basically the Mercedes logo without the ring. So looking at shorter and longer branches. So this is a very long branch, and the density really goes down from the middle to the end. And still with quite a bit of noise, you keep seeing the same cluster. Whereas with HDB scan, this would all be one cluster anyway, right? HDB scan would be able to identify the different clusters if the density was the other way around. So if the middle was the least dense, then it would find 3 clusters, but not if the middle is the most dense. Scalability, I'll skip that. So that's Flask for identifying flares. We also wanted to find an easier way to actually reproduce the work done by Peklum on this breast cancer data. So, because the algorithm that they use, that mapper algorithm, one with the hand, has a lot of different parameters. So it's quite involved in actually getting things out. So we want to make it as easy as possible. And that's why Jelmer developed the lensed UMAP. So if you have UMAP algorithm and you look at ESR1, so again, these nodes are different patients and they're colored by the expression value of ESR1. I see this as rainbow colors. You didn't see this, right? So colored by ESR1. Actually, if we use a lens, for example, the L infinity centrality, so how different are my points from other points, then we really immediately get a network where these points are very different from all the other points, and these are indeed the patients that are separate. So this calculates like the Vietores-Rips complex in the background, just to basically get to that manifold approximation. There's different ways we can actually apply such a lens. So yeah, so how do we do this? There's different ways. One is that you can, for example, see you create a lens across your dimension that you want to use as a lens, right? And you bin, but not with overlaps. And then you basically generate your UMAP network and remove all the edges that are not within one bin or between 2 adjacent bins. So if they are too far, like the light gray ones, you remove those. Only within the same bin or with an adjacent bin, you keep those. That's what we call the global lens. Then there's a global mask and a local mask. Basically, the global mask, it creates two networks. Basically, the general UMAP network, and then looking at the K nearest neighbor network, and then basically just does an intersection between the two. And the local mask looks at every single point individually, like which of the UMAP links are also are actually links to the K nearest neighbors. That goes a bit into technical parts. I'm basically looking at the same information again from the breast cancer patients. So each node here is a breast cancer patient. Just regular UMAP, we find this subgroup here. And if you look at what is the difference between the big group and the small group, well, that is the ESR1. So if we color by ESR1, yes, we see a much lower expression of that gene than in the rest. Within this group, there's still people who are who get a relapse and people who do not get a relapse. And we looked at that, and then we basically see there's another gene that actually plays a role there as well. But I guess quite into the domain itself. A final thing on the topology part is something very, very recent. It's one of those examples that you think afterwards, like, okay, we spent like a decade on this, but actually there's a much simpler way to do this. To build a tree, or to build that network, so I showed you that stat network, making the distance matrix, and then really adding edges and comparing, well, calculating correlation between the different distance matrices. We basically found out after discussing with people from TU Eindhoven in the Netherlands that if we just create a minimal spanning tree, well, I'll first go over the noisy MST one. You build a minimal spanning tree and everything is connected to everything else, okay? But that's not the complete shape yet. So imagine you have your data is actually in a circle. With a minimal spanning tree, by definition, you will not have a circle. But what you can do is actually you can add noise. So you have your initial distance matrix, you build a minimal spanning tree. Then you could add noise to that matrix, build and calculate the second minimal spanning tree. Add additional noise, basically just changing the values a little bit in the distance matrix, you get a third minimal spanning tree. In the end, if you just collapse all those minimal spanning trees, you get quite a good network. That's one way of doing it. Another way of doing it is in the Birovka algorithm for calculating minimal spanning trees, where we changed the algorithm itself a little bit. Instead of picking the closest nodes to connect to build a minimal spanning tree, it may connect to a node that's a little bit further off. It might bypass the first one. So that's very similar ways of actually doing the same thing. But these are some preliminary results of that. So let's say you have a point cloud that looks like this. And importantly, there's a gap here. If we run UMAP with two nearest neighbors, then we have a lot of connected components. This is not one cluster yet. Right? So a lot of disconnected components. And if we have 5 nearest neighbors, only at that point we can close the gap here, but also there's this link that should not be there. Whereas if we run our methods with just two nearest neighbors, then immediately that gap is closed, so we get the manifold. The same thing with a horse data set that we very often use. So this is a 3D point cloud of a horse. With our methods, we very quickly get the shape of the horse back. But with UMAP, it's much more involved. And you get the moment that you get one connected components to get one horse, there's a lot of links there that should not be there. So it doesn't look like a horse anyway. To wrap up, so basically the final thing I want to mention is like also here. So we work with topological data analysis, which is basically network based, but sometimes I want to actually look at multiple networks at the same time. So in biology, it could be genomics, proteomics, or metabolomics. But specifically in topological data analysis, imagine you have patients where you calculate distances between patients based on blood values, white blood cells, red blood cells, lymphocytes data, and so on. So you get this network, the blue one, but also there might be other information, and you get a different network for the same patients. How can you actually start analyzing that? So I will have a PhD student starting in September actually looks into visual analytics of multi-layer networks. So basically, I hope that gives a little bit of an idea of what we try to do. So we try to get people so far that both experts and lay individuals that they can embrace complexity again. Instead of throwing complexity away, instead of throwing outliers away, instead of looking only at the limited number of dimensions, embrace that complexity. That's as much a philosophical question as a technical or a research one. To look at uncertainty, that's something else that's very difficult to get people to think about. If you think about the models, infection models during COVID, that was not easy, right? So of course, this is not work that I did myself. So as a professor, you don't do anything yourself. These are the people who did that work over the years, and with collaborations as well. So I really want to thank those people. And with that, I thank you for the introduction and the opportunity to talk to you as well. So if you have any questions, I'd be happy to hear them.
Thank you very much, Jan.
Yeah.
All right, we have time for a few questions. Who wants to go first? Yeah,
I maybe have a brief question regarding the flare-sensitive ICG scan project. I was just wondering whether the code for this is somewhere like publicly available to try it out.
So, say again, so if the code is available?
Yeah, for the flare-sensitive ICG scan.
Yes, that's all available, and I'll share the GitHub repositories of those 3 projects with you after the talk.
Amazing, thank you.
Yeah. By the way, so both Flask and like the lensed UMAP, we are in talks with Leonard McInnes to actually get that into the Python implementations as well of in scikit-learn. So eventually they might end up there.
Cool, yeah. Thank you.
Okay, more questions?
By the way, this is this is topological data analysis of climate data that you see now in the background.
Maybe I can continue that. So Jan, there is this discussion of, if you look at topological data analysis, you do you basically operate in the down projected space, right? Like
Yeah.
How does this relate to approaches that work in the original high dimensional data space? Because ultimately you lose information. So are there clusters that can then be found in the original high dimensional space? And like, what are situations where you would apply the one over the other? Basically,
very often we end up just doing a topological data analysis. So in any analysis we do, well, it's always something for an expert, and it always ends up being like a dashboard looking at different views at the same time. And the topological data analysis often, that network is very often like the overview of everything. So you basically select certain points, certain patterns in that topological data analysis, and then you go from there, right? Yes, there will be patterns that you would find in the high dimensional space itself. But basically, we stay at the topological level mainly. In a sense, it's more interesting because you can look at the data from different views as well. Like I mentioned, using different lenses and so on. So looking at the data from a certain perspective, looking at it from a different perspective, looking at different perspective, and actually combining those into one, that can give you a very, very fine-grained idea of subpopulations in the data and trends and patterns. Does that answer your question a little bit?
So that would be your multi-layer approach that you mentioned at the end.
Yes. At this moment, it's a bit hacky what we do to do this. It's a bit ad hoc, but actually to find a good multi-layer way of exploring complex data, that would be useful.
Mm-hmm. And could you go back to the slide where you had this the approach where you added noise and then did the topological analysis again?
This one?
No.
Or
yeah.
This one? But
yes. So you mentioned that you add noise and then you have different topological or different spanning trees that you collapse. What do you mean collapsing?
You basically just add them to each other. So basically, if you let's say you have 4 points, and normally I now open something to draw, but let's say you have 4 points, right? And based on your distance matrix, you connect the 2 at the top, the 2 at the bottom, and then the 2 on the right, right? I don't know if that's your or my right. So you have the top, right, bottom. If you then change the values a little bit in that distance matrix, it may be that the 2 on the left are closer together than the 2 on the right, which means you have a new minimal spanning tree that is at the top and on the left and the right and then the bottom, right? So one spanning tree is a top, right, bottom, another one is top, left, bottom. In the end, we just collapse them, which means you have the top, left, bottom, and right. You just add them together.
Okay.
That's the only thing.
All right. Yeah, thank you.
I would like to ask a question. It might be it's about the clusters which you are working with. So I didn't quite become sure whether those clusters are kind of linked together in the very beginning always, or if we are talking about clusters which were created by some kind of cluster finding algorithm like k-nearest neighbors.
You know, like for example, in this slide here, or
somewhere you said you are using using an overlap between k-nearest neighbors and the other algorithm. I think it was somewhere at the end.
Was it this one?
I think so.
Yeah, so basically with the network that we built, by definition, yeah, so one thing, by definition, they are one big connected component. Everything's connected to everything else because we start from a minimal spanning tree. That's one thing. A second thing, yes, clusters. You can identify those by looking at community detection algorithms, for example. That's something that we can do. But in this case, so here what we do is so the K and the K nearest neighbors networks is basically we create the network from UMAP. So the underlying network that UMAP uses, and separate from that, we do a K nearest neighbor network,
which is not map, not.
That's not UMAP.
The UMAP stat.
No, that's just looking at that's just looking in high dimensional space. What are the 5 points that are closest by?
Okay,
period.
And the other network is created after UMAP.
Yeah, and then they would just look at the intersection between the two.
Okay,
that's it.
Because I wasn't sure how there is a difference because the other clustering might as well be k-nearest neighbor.
All right, yes. No, it's just one K nearest neighbor with looking at the well, making a full K nearest neighbor network. So the global mass is making a full K nearest neighbor network and doing the intersection with the UMAP network. And the local lens is really just looking at every single point individually. But that there are minor differences that do have some effects on the use, though. So here with the local mask, you might end up splitting into multiple components where that's not really the case with the global mask, for example. But here the compute cost is really high. But we're still working on this as well, so this is not finished work. It's still in the process.
Okay, thank you.
Thank you. One final burning question. If not, then thanks a lot, Jan, for this great talk again. Thank you. And so what remains open is that I announce the next talk, which is going to be held by Michael Erich. He's a professor at Utrecht University, and it's going to be at the 22nd of May, again at 2 p.m. Summertime. Thanks everyone for joining, and see you soon. Bye. Have a great day, Jan. Thank you.
Okay. Thank you, Mark. Bye-bye.
Bye-bye.